4.1 is a photograph of a tree that has been girdled. Girdling involves removing a complete circle of bark and phloem from around the tree. I. Explain why the area above the girdle in Fig. 4.1 will become swollen. Use the term source and sink in your answer. Any four from reference to translocation, carbohydrates, glucose sugars, amino acids, made in the leaves or reference to photosynthesis in leaves. 3. Glucose carbohydrates, converted to sucrose. 4. Leaves act as a source and roots act as a sink. 5. Phloem transports sucrose amino acids. 6. Sucrose, named sugars, collect above the girdle. 2. Explain why the leaves in girdled trees are still able to receive mineral ions from the roots. Xylem is still intact idea that xylem is further inside the trunk. Mineral ions transported by xylem. B. Explain why glucose made during photosynthesis is required for the absorption of mineral ions by the roots. Any 3 from idea that glucose required for aerobic respiration to release energy for ATP production. Reference. To active transport, to transport mineral ions from low to high concentration against their concentration gradient. C. State the balance symbol equation for photosynthesis. 0, 610 underscore M22 underscore MS underscore 42. 3a. A scientist investigated the effect of temperature on the mass of leaves picked from a tea plant, Camellia sinensis. 3 samples of leaves were picked and the mass of each sample of leaves was recorded. Each sample of leaves was kept at a different temperature for 4 hours. After 4 hours, the mass of each sample of leaves was measured and recorded. Again. The scientist then calculated the final mass as a percentage of the initial mass for each sample. Fig. 3.1 shows the results. I. Explain the results shown in Fig. 3.1. Any 5 from. As temperature increases, the mass of leaves decreases / reference. By increased transpiration. Evaption increases. From surface of the mesophyll cells. Due to increased kinetic energy of water molecules. Diffusion of water vapor increases. Through stomata. 2. State one factor, other than temperature, that would affect the loss of mass from the leaves of a plant. Humidity. Reference 1, for example, wind speed. B. Fig. 3.2 is a photomicrograph of the tissue that transports water and mineral ions in a plant. I. State the name of the tissue shown in Fig. 3.2. Xylem 1. 2. Describe how the tissue shown in Fig. 3.2 is adapted for its functions in the plant. Any 3 from hollow, no cell contents. To reduce resistance, allow efficient flow. No end walls to reduce resistance, allow efficient flow. Large cross-sectional area wide to allow transport of large volume of water. Lignin 2. Prevent collapse, provide support, provide strength. Waterproof to prevent water loss. Reference, for example, bordered pits for lateral fluid transport. C. Explain how mineral ions enter a plant. Any 3 from into the root hair cell by active transport against a concentration gradient / reference, using energy, using carrier proteins in the cell membrane, also initially by diffusion. 0, 610 underscore W22 underscore MS. Underscore 4, extended 3, 4a, Fig. 4.1 shows a dandelion plant, Taraxacum officinale, in a field. The flower stalk is called a scape. I. The scape of a dandelion responds to sunlight by growing upwards. State the name of this growth response. Positive phototropism. 2. The scapes of dandelions keep the plant upright without the need for structures such as bones. Explain how cells in plant scapes and stems keep plants upright. Any 2 from plants have cell walls. Pressure of water pressing against cell wall reference. To turgor? Reference. To xylem offering support? Reference to lignin. B. Fig. 4.2 is part of a cross-section through the scape of a dandelion. B. Fig. 4.2 is part of a cross-section through the scape of a dandelion. Draw a line and add a label on Fig. 4.2 to identify one area of xylem tissue. Label to xylem shown on Fig. 4.2 C. Describe the function of phloem tissue. Any three from translocation, transport of sucrose amino acids. Sugars transports nutrients from named source to named sink. Reference, for example. Transport can occur in both directions. Some organs can be both a source and/or a sink at different times. Note: MP4. For example, transport can occur in both directions. Some organs can be both a source and/or a sink at different times. D. A dandelion scape was cut into long strips for an osmosis investigation. Immediately after the scape was cut, the pieces of scape bent outwards, as shown in Fig. 4.3. Using the information in Fig. 4.3 and Fig. 4.4, deduce and explain which strip of scape was in the most concentrated salt solution. Strip of scape. Explanation. Strip of stock. B. Explanation. Max 2 from. Shows greater degree of bending, shriveling / reference because more water moves out of the cells in the inner surface of the scape. Water moves from high water potential to lowest water potential, lower water potential. Cells become plasmolyzed, flaccid, less turgid. 0610 underscore S23 underscore MS underscore 42 1 Fig. 1.1 is a diagram of part of a cross-section of a leaf. A. Explain why a leaf is considered to be an organ. It is made of named tissues in a leaf. Tissues, working together for a specific named functions. B. Explain how layer B and layer C in Fig. 1.1 are adapted for their functions. Ref to photosynthesis. B is the palisade, mesophyll tissue layer cells, and C contains spongy, mesophyll tissue layer cells, vascular, bundles, xylem phloem. Any three from layer B. Cells are tightly vertically packed or columnar or few cross walls. Cells layer positioned immediately below. Layer A, the upper, epidermis to maximize light, energy. Reaching cells chloroplasts layer, ref, to many chloroplasts, lots of chlorophyll. Large vacuole keeps the chloroplast towards the sides of cells. Any three from layer C, cells are loosely packed. Layer contains interconnecting air spaces between cells, mesophyll air spaces, to allow gas exchange diffusion of named gases, mesophyll air spaces, to allow evaporation of water from cell surfaces into air spaces. Xylem has thick lignified waterproofed pitted cell wall, xylem, long, continuous tube, no cytoplasm hollow or no end cross, walls, xylem, to allow transport of water mineral ions, phloem, to allow translocation transport of sucrose sugar amino acids. C. A student placed a plant in a very hot room for 12 hours. There was a bright light in the room, and the plant was not given any water during the 12-hour period. Fig. 1.2 shows a series of sketches that the student made of the stomata during the IE2 investigation. I. Identified the cell labeled D on Fig. 1.2. Guard cell 1. 2. State the main function of the stomata. Gas exchange described regulation of transpiration / reference. 3. State the advantage to the plant of the change to the stomata shown in Fig. 1.2. Reduce water loss through transpiration. Reference to preventing wilting. D. The student increased the humidity in the room and repeated the investigation. Predict and explain the effect of high humidity on the stomata. Any three from Boigreau. Stomata will take longer to close, reduces stomatal closure, reduces stomatal response. Reduced, water vapor concentration diffusion, gradient between the air spaces and the outside. So the guard cells do not lose water as quickly much as the first experiment. The guard cells become Turgid or guard cells take longer to become flaccid. 6a, c. A student investigated plant growth responses in roots and shoots. They used this method. Damp cotton wool was placed in 2 petri dishes. 3 bean seedlings were attached to the cotton wool in each petri dish. Each seedling was orientated so that the roots pointed in a different direction in each petri dish. Petri dish 1 was kept on its side in a fixed position. Petri dish 2 was kept on its side and rotated constantly. Both petri dishes were kept in the dark. Both petri dishes were kept in these conditions for 2 days. After 2 days, the seedlings were observed. Fig. 6.1 is a diagram of the apparatus. Fig. 6.2 shows the seedlings after 2 days. I. Describe the pattern of growth of the bean roots and shoots in petri dish 1 shown in Fig. 6.2. Either all roots have grown downwards with gravity no matter which way they were facing at the start, or roots show positive gravitropism geotropism, and either all shoots have grown upwards in opposite direction to gravity no matter which way they were facing at the start, or shoots show negative gravitropism geotropism. 2. State the name of the growth responses observed in the bean roots and shoots. Gravitropism. 1. Way. Explain how auxin causes the difference in the pattern of growth shown by the shoots of seedlings B and E shown in Fig. 6.2. NE5 from Sora, auxin maiden released from shoot tip. Auxin moves by diffusion. Auxin stimulates cell elongation. In B petri dish. 1. Auxin has a higher concentration of auxin on the lower surface of the shoot. Auxin stimulates cell elongation more on lower side of the shoot. Shoot grows away from the pull of gravity/reference. In E petri dish 2, auxin is equally distributed in the shoot. Auxin stimulates cell elongation equally on all sides of the shoot. Shoots grow neither up or down. Shoot grows straight because the effect of gravity is constantly changing, countered by rotating the petri dish. B. Seeds require oxygen and water to germinate. I. State one other environmental condition that affects germination. Example of an appropriate environment condition. 1. For example, temperature. 2. Suggest why oxygen and water are required for germination. Any 2 from oxygen aerobic respiration. Water used in named cell process. Water is a solvent/reference. Water used for turgor, reference 0610_S23_EMS_43. 2. Transpiration is the loss of water vapor from the leaves of a plant. A. Complete the sentence describing transpiration. Water evaporates from the surfaces of the mesophyll cells into the air, spaces, and then diffuses out of the leaves through the stomata as water vapor. B. Explain how water moves upwards in the xylem. Any 3 from transpiration pull draws up a column of water held together by forces of attraction between water molecules. Xylem forms a continuous empty tube. Loss of water from leaf reduces water potential hydrostatic pressure. Reference, for example, adhesion between water molecules and wall of xylem/reference. C. A student investigated the effect of wind speed on the rate of transpiration in a small Pachira aquatica tree dot. They measured the rate of transpiration when the plant was placed in different wind speeds in both the light and the dark. The tree was given an adequate supply of water. The results of this investigation are shown in Fig. 2.1. I. Describe and explain the results shown in Fig. 2.1. Describe Max 3 from. As wind speed increases, the rate of transpiration increases in both light and dark. The rate of transpiration is higher in the light than the dark. The rate of increase in transpiration rate is higher in the light than the dark. Reference change of gradient at wind speeds above 4 m per s to 10 m per s. Comparative correct data, quote, including units. Explain, transpiration rate increases because the water vapor is removed from outside the leaf faster/reference. maintaining the concentration gradient for diffusion. In the light, more stomata are open wider, to allow gas exchange carbon dioxide to diffuse and oxygen to diffuse out. For photosynthesis, energy from light increases the kinetic energy of the water, molecules rate of evaporation of water. 2. The investigation was repeated in the light with the same species of tree in an environment where the humidity was higher. Draw a line on Fig. 2.1 to predict the results in the light with increased humidity. 1. Line drawn increasing with wind speed but below the light line. One must start below 2 g per hour. A below the dark line. 3. The investigation described in 2c was repeated, different wind speeds in both the light and the dark. However, the tree was not given an adequate supply of water during the investigation. Predict the result and explain your prediction. Any 4 from prediction. Lower rate of less, transpiration in the light, at all wind speeds, little change in the dark. Explanation. Max 3 from less water absorbed by the roots, water moves from the cells by osmosis. Guard cells lose their turgidity, become flaccid. In the light, stomata close, plant leaves tree, wilts. Little change in dark because stomata already closed. 0, 610 underscore W22 underscore MS underscore 4 extended 1. 3a. Fig. 3.1 shows some apparatus that was used to investigate water loss from a leafy shoot. I. State the name of the process by which leafy shoots lose water. Transpiration 1. 2. Before the leafy shoot is inserted into the jar shown in Fig. 3.1 comma, it must be recut under water. Suggest why the end of the leafy shoot was cut under water. Ensure a continuous column of water, prevents air bubbles, prevents airlock. 3. State the purpose of the oil layer on top of the water in the burette. Prevent evaporation, condensation from the top of the burette, affecting the volume of water in the burette / reference. IV, using the information in Fig. 3.1 comma, describe one method that can be used to determine how much water is lost from the leafy shoot. Measure the decrease in the volume of water in burette over a period of time, record the decrease in mass over time. B. The apparatus shown in Fig. 3.1 was used to investigate the effect of temperature on the rate of water loss in a species of plant. The results are shown in Fig. 3.2. I. Using the information in Fig. 3.2 comma, calculate how much water would be lost from 1 square meter of leaves in 12 hours if the plants were kept at 35 degrees Celsius. Include the unit 12096 g per square meter, 12.096 kilograms per square meter, a 12.1 kilograms per square meter. MP1 correct reading from graph 0.28 GT per square meter per S. MP2 correct calculation 0.286060 12.12096 G. MP3 correct unit G or kg. ECF for MP2 from incorrect MP1. 2. Using the information in Fig. 3.2 comma, describe and explain the effect of increasing temperature on the rate of water loss in this species of plant. Any 5 from 1. As temperature increases, rate of water loss increases. 2. Reference to steeper rate greater loss of water after 3738 OC. 3. Any correct comparative data quote with units at least. Once. 4. Water vapor lost through stomata between guard cells. 5. Evaporation from mesophil into air spaces. 6. Diffusion down. A. Water potential gradient. 7. Increasing temperature increases kinetic energy of water, molecule. 8. Faster rate of more. Diffusion. 9. Stomata open wider. More stomata open and higher. Temperatures. 10. Reference. C. The apparatus shown in Fig. 3.1 can also be used to investigate the effects of changing humidity on water loss in plants. I. Suggest why the mass of water in the apparatus does not change when the leafy shoot is kept at 100% relative humidity. No diffusion of water vapor, because no water potential gradient described. 2. Even at extremely low relative humidities, the leafy shoot did not wilt. Explain why the leafy shoot shown in Fig. 3.1 did not wilt. It has a continuous supply of water/reference. 3. The investigation on the effect of temperature was done at a relative humidity of 20%. The investigation was repeated at a relative humidity of 80% and all other conditions were kept the same. Predict how the water loss will differ from the trend shown in Fig. 3.2. Sketch your prediction on Fig. 3.2. Line drawn below original line 0610 underscore W19 underscore MS underscore 42 2 Fig. 2.1 shows part of a cross-section of the stem of a young sunflower plant. 2a, any shape drawn that includes one whole vascular bundle, including all of the xylem and phloem with or without sclerenchyma. Label line from X to xylem in any of the vascular bundles. A. Draw a circle around one vascular bundle on Fig. 2.1. Label the xylem in the vascular bundle with the letter X. Any shape drawn that includes one whole vascular bundle, including all of the xylem and phloem with or without sclerenchyma, label line from X to xylem in any of the vascular bundles. B. Explain how the cells in area Y are able to support the stem so that it stays upright. Cell vacuole cells contain much water, have high water potential. Water absorbed by osmosis down water potential gradient. Cells are turgid, have a turgor pressure. Cell contents vacuole cell membrane, pushes out against cell wall. Cell wall does not stretch, is inelastic, is rigid. Reference, for example, cells are tightly packed, /reference. C. Researchers used carbon dioxide that contained a traceable source of carbon, 13C, to investigate translocation of sucrose from the leaves of bean plants, Phaseolus vulgaris. Fig. 2.2 shows that glucose produced in photosynthesis is converted to sucrose for translocation. Researchers selected 4 plants, Q, R, S, and T, which had leaves that were of similar sizes. The leaves on the 4 plants were supplied with 13CO2. After the leaves had started to make sucrose, the researchers cut away a ring of tissue in different places as shown in Fig. 2.3. The rings of tissue that were removed from plants R, S, and T contain the phloem. The quantities of sucrose containing 13C in the shoot tips and in the roots were determined. The results are shown in Table 2.1. Describe and explain the effect of removing the phloem on the translocation of sucrose in plants Q, R, S, and T. Q, sucrose 13C is in shoot and root. Tino, sucrose 13C in shoot or root. R, sucrose 13C in root only in root but not in shoot. S, sucrose 13C in shoot only in shoot but not in root. Idea that no transport of sucrose 13C where phloem is removed. Phloem transports sucrose in both directions. Leaf is source carbon dioxide is fixed in leaf. Sucrose is made in leaf. Roots shoots are sinks described. For example, of descriptions respired stored as starch converted to another, named compound. Reference EGQ is a control. 0610 underscore S20 underscore MS underscore 42 6A Fig. 6.1 is a diagram showing some parts of a plant. The circle shows a magnified cross-section of part of the stem. I. Table 6.1 contains statements about the functions of some of the structures in Fig. 6.1. Complete the table by stating the name of the structure, identifying the letter that labels that structure. 2. State one letter from Fig. 6.1 that identifies a structure that contains a haploid nucleus. B. DF1. The. State the name of the process that describes the transport of sucrose in a plant. Translocation 1. IV. State one letter from Fig. 6.1 that is a structure that is an example of a source for sucrose transport. H. 1. B. In addition to sucrose, amino acids are also transported in plants. I. State the name of a mineral ion that becomes part of an amino acid. Nitrate ions. 2. State the name of the structures inside cells that assemble amino acids into proteins. Ribosomes, rough endoplasmic reticulum. 1. 3. State the name of the group of molecules that a